In this video, we will be presenting the procedure to conduct our experiment for selected environmental parameters as part of our environmental engineering laboratory mini project. Our group consists Sharifah, Fatimah, SALBIAH, Wan Nur Amirah, Nur AINI SYAKILLA, Muhammad HAKIMI, and RAYMOND ANAK TIKOK. The first test is total coliform. The apparatus starts with Coliland region, quantity tray 2000 tray with 97 wells, region bottles, UV lamp 365 nanometer used to detect fluorescence from E. coli, quantity tray sealer plus incubator with incubation temperature of 35 degrees Celsius. 110 ml water sample was collected in a glass region bottle and mixed with Coliland region, then gently shaken until fully dissolved. The prepared mixture was poured into a quantity tray which was then sealed using the quantity tray sealer plus to create 97 individual wells. The sealed tray was incubated at 35 degrees Celsius for 24 hours. After incubation, the tray was examined for color changes. Wells that turn yellow indicate the presence of total coliform, while fluorescent wells observed under 365 nanometer UV light confirm the presence of E. coli. The number of positive wells was counted and the most probable number MPN per 100 ml was determined using the IDEX MPN table.
Next, we have ammonia test. For the apparatus using in this test is first we have one set of ammonia nitrogen reagent, and then we must prepare 25 milliliter of ionized water. Next, we have 2 of graduated mixing cylinders, and then we have sample cells 1 each square for 10 milliliter for 2 samples, and lastly we have 2 of serological pipette for 1 milliliter each. And then for the procedure for this test, firstly we must prepare a sample which is fill a 25 milliliter mixing graduated cylinder to the 25 milliliter mark with the sample. For the next step is add 3 drops of mineral stabilizer to each cylinder, stopper and invert the cylinder several times to mix. And next we must add 3 drops of polyvinyl alcohol dispensing agent to each cylinder, stop and invert several times to mix. And then we have 1 milliliter of Nestle reagent into each cylinder, stopper and invert several times to mix. And then set time for 1 minute's reaction period to begin. After that, we must pour 10 milliliter of each solution into a square sample cell. And then, when the timer expires, insert the blank into the cell holder with the fill line facing right. Press 0. The display will show 0.00 milligram per liter. And then, write the prepared sample and insert it into the cell holder with the fill line facing right. Lastly, press read and take the result in a milligram per liter.
Chemical oxygen demand test. The apparatus that we need is COD wire, pipette, spectrophotometer and beaker. Firstly, pour the water sample into the beaker, and then by using the pipette, take 2 ml of the water sample and pour it into the COD wire by using the low range. This is because the water sample is from the lake, and then inverted the wire until it dissolves. After that, the mixture is placed at the COD reactor with the temperature 150 Celsius for 120 minutes. After 120 minutes, cool down the mixture until 120 Celsius, then take out the sample. Finally, by using the COD low range program, we take the reading for both sample and blank. Repeat the last step for 3 times to get the more accurate reading.
Next is BOD task. The apparatus needed are BOD bottle of 300 ml, measuring cylinder, DO meter, distilled water, incubator at 20 degrees Celsius, dilution water that contain phosphate buffer, magnesium sulfate solution, calcium chloride solution, and also ferric chloride solution. Firstly, we need to prepare a dilution water where it is a combination of distilled water or deionized water with phosphate buffer, magnesium sulfate solution,
calcium chloride solution and ferric chloride solution, where the solution will be aerated for at least 2 hours to ensure that it is saturated with dissolved oxygen. Then measure the water sample with a measuring cylinder for 10 ml and pour it into the BOD bottle. Then fill up the BOD bottle with dilution water until the neck of the bottle. Close the BOD bottle with its cap and turn the bottle upside down to make sure that there are no bubbles. Then use the DO meter to measure the dissolved oxygen as DO0. Make sure that the end of the DO meter is clean with distilled water before every use. Take 3 readings to get the average of DO0. Then fill up the BOD bottle again with dilution water to avoid air pockets inside the bottle. Put the BOD bottle inside the incubator at 20 degrees Celsius for 5 days. After 5 days, repeat the same process by using the DO meter and take 3 readings to get the average value of DO5. Lastly, by using the formula of average DO0 minus average DO5 divided by decimal volumetric fraction of sample used, we will get BOD5. With the value of BOD5, compare the result with the Malaysian standard.
Suspended solid test. The apparatus that we need is beaker, blender and spectrophotometer. Measure 500 ml of the water sample and then pour it into the blender. And then blend the 500 ml of water sample by using the high speed for exactly 2 minutes. And then pour the blended water sample into the 600 ml of beaker. After that, immediately pour 10 ml of blended sample into a spectrophotometer and also prepare the second sample with a 10 ml of the tap water. After that, choose 630 suspended solid program to take the reading for both samples and blank. Repeat the last step for 3 times to get the more accurate data.
Next, pH test. Apparatus, pH meter, clean container or beaker, distill water for rinsing. First, prepare the water samples. Second, rinse the pH probe with distilled water to avoid contamination. Third, gently dry the probe using a tissue. Fourth, submerge the probe into the water sample. Fifth step, wait for the reading to stabilize on the digital display. Sixth, record the pH value directly from the screen, and after use, rinse the probe again and store it properly.